Welcome to the Jack Westin MCAT Podcast with your hosts, Michael Maleka
and Molly Hudash. Well, hello, everyone. Welcome back to the Jack Westin MCAT Podcast with your hosts, Mike and Molly. Mike, we always have high yield material to cover. That's kind of the point. But I feel like this episode is pretty high yield.
Right? I think we both love this topic today because of, one, how important it is, like on the topic, but it's also really interesting. I think it is so cool in this topic, and it really builds off well what we talked about last episode when it comes off thermodynamics, right? Last episode, we covered thermodynamics. We talked about how it relates to electrochemistry. It was a jam-packed episode, actually. Like when I kind of reflect on it, we covered so much last episode. So if you haven't checked it out, go check it out. We talk about thermodynamics. We built really off redox reactions, and then we tied it all together for like batteries, galvanic cells, electrolytic cells. But today we're kind of doing the other side of the coin, kind of the exact opposite of thermodynamics. Again, we want to make this very clear. What we're talking about today is very different from thermodynamics. Molly, what are we talking about today?
We're talking about kinetics.
Kinetics,
and I know we set it up last episode, right? These two concepts, they're brought up together a lot. Students confuse them a lot. They are completely separate disciplines. They're separate worlds, right? Different planets that these are on. Thermodynamics, like we mentioned, is all about examining the energy or the stability of products and reactants. We're asking and answering the question, will this reaction happen and therefore be spontaneous if it's energetically favorable? We're producing things that are more stable than we started with, right? Yes, that will happen, or maybe it won't happen. Maybe we need an input of energy to invest in our product or our reactants to make products, right? Thermodynamics, will a reaction happen based on the stability or the energy of both products and reactants? It's very reflective. We talk about things like KEQ, delta G, delta H, delta S, temperature, all that good stuff. In this realm or this world, in kinetics, we ignore all of that. We assume that the reaction is going to happen. Assuming the reaction happens, how fast can I get this to go?
All about the speed. We are all about speed. Reactions, products, we don't care about those ones. But like, how fast is this reaction going? And it's kind of like the midway in between the two. How do we get there? Right?
Yes.
And so that's going to be talking about today. And I think a good place to start talking about this is when it comes to K. That letter. I don't know why in chemistry they love that letter, right? And because there are so many K constants, but we've talked about some really important K constants before as it relates to equilibrium. We had a whole episode on equilibrium homeostasis. If you haven't checked out that episode, go watch it. But when we talk about the K in equilibrium, we talk about capital K or big K. It's about related to the reactants and products. And capital K, though, is what kind of a constant, Molly? Is it a kinetic constant?
No. Well, that's a loaded question, actually.
That is a loaded question. That is a very loaded
question. Big K, little subscript. In every case except one, we're talking about an equilibrium constant. So KEQ, that's just the general equilibrium constant. We also, remember, talked about this with acids and bases, Ka, Kw, Kb, right? We also talk about things like Ksp, Kc, Kd, all of this, right? There's so many K constants. They are all equilibrium constants. They live in thermodynamics world.
They're all thermodynamic constants. Absolutely.
There's one exception.
We'll talk about it today. We'll talk about it today. It's a little teaser. There is one exception, but most of the time, if you see a capital K, right? I kind of want to leave it. And a little subscript, except the one which we'll I want to leave it as a cliffhanger for when we do talk about it. Is a thermodynamic constant. It's an equilibrium constant, but this is where we have to introduce the idea of little k. Underscore K. And I don't know why. They could have chosen any other letter. Why did they choose K? But if you see a capital K, think equilibrium constant, except in that one case. If you see a lowercase k, we are now in the other world. We are now in rate land, because this is our rate constant.
Yes.
Now, our rate constant is related to the kinetics of a reaction. And now the rate constant, when we think about the rate constant, there's an equation we think of that you'll never have to, I don't think I've ever actually seen a problem that actually has you to calculate using this equation, but we should know it conceptually, which is the Arrhenius equation. Now, again, maybe I'm asking a little bit much of my editors, but if the editors are nice enough, they can put it on screen. If for those watching about the Arrhenius equation, it kind of looks like a little bit of a scary equation. I'm going to be honest, but again, I'm going to maybe just say it verbally. It's lowercase k, little k equals capital A, the Arrhenius constant, multiplied by Euler's constant, which is an E, to the exponent of negative activation energy over RT. Now notice, that was a mouthful. That was a mouthful.
So many people glazed over.
You're like, Mike, you just said an equation. If it's on screen, great, look at screen. If not, you can always look it up. Thank you. That's the Arrhenius equation. But it's this Arrhenius equation that tells us what factors or what variables impact the rate constant. Now, when we think about rate, there's generally 3 variables we think of, but 2 of them are described in this equation. In this equation, the 2 variables that impact the rate constant are 1, the activation energy of a reaction, and as well the temperature of a reaction. So I kind of want to first start about temperature, because temperature is kind of a really cool factor when it comes to reactions, because temperature, we've talked about before, can impact the equilibrium of a reaction, but it also impacts the kinetics of reaction, where I think temperature is so cool in that sense, because when it comes to equilibrium, we know temperature can shift the equilibrium, but how would we know if temperature, how do we know the direction, Molly, of how temperature is going to shift the equilibrium?
Well, you would use your delta G equals negative RT ln KEQ.
I love that.
I'm like, wait, I know this one.
Yes, I know. I'm kind of putting you on the spot. I know. But yeah, where temperature can impact the direction a reaction will go. And we actually know temperature can impact whether the products or reactants it shifts towards them. And we can use our ideas of delta H that we went and talked about last episode. Because we talked about last episode, the ideas of something being exothermic versus endothermic. Do we need heat or do we produce heat? But whenever I think about how temperature impacts equilibrium, I like thinking about is temperature a reactant or is it a product?
Yeah.
Right. And so then you can just kind of introduce the ideas of Le Chatelier's, right? Oh, if I'm increasing the reactants, is going forward. If I'm increasing the products, I'm going backwards. If you hear me say the word Le Chatelier in my Frenchish accent as a Canadian, and you're like, what is he talking about? Go watch that episode on equilibrium and homeostasis. We talk all about Le Chatelier's. So we know temperature can shift the equilibrium, but it can also impact the kinetics of a reaction. And so while depending on every reaction, it shifts right, shifts left if we increase the temperature, but always is the case. If you ever increase the temperature, you always increase the rate, no matter the reaction. You increase the temperature, you make the reaction go faster. Because imagine, temperature is just the average energy in the environment. If you increase the amount of energy, I'm like, that's like me on caffeine. I'm super hyper. I'm like moving around these things. More reactions are likely to happen. You're going to make these reactions go faster. So always increasing the temperature is going to increase the rate.
I feel like it's an important point because you're saying if you hot me up on caffeine, I could do anything. When it's inorganic, yes. When we talk biological applications, which we will later with enzymes, there is a Goldilocks zone. And it's the same way with you. If you were on a ton of caffeine, you get to a point where you can't do anything anymore. Okay. When we start to heat up biological systems infinitely, we're going to have a problem. But theoretically, in an inorganic Gen Chem sense, more heat, faster reaction.
Absolutely.
Period. High temperature, faster, 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 faster. And like you said, it will shift KEQ based on whether heat is a reactant or a product. But I always want to add that. I'm like,
okay, don't go on the surface of the
yourself thinking that it'll make you more productive.
Please don't do that.
Don't try this at home.
Preface, please don't try that at home. But temperature is cool because it impacts both the thermodynamics and the kinetics. Kind of both these two worlds, temperature does both. But then the Arrhenius also talks about another variable that impacts the rate of a reaction, and one that's very, very important, which is the activation energy. Now, Molly, walk me through what is this activation energy that's related to kinetics but not thermodynamics?
Yeah, so activation energy is basically, you know that saying, you have to have money to make money, or you have to spend money to make money?
Yeah, absolutely.
See, that's a very common phrase, and I feel like it really explains well what activation energy is, right? If we think about going back to our earlier episode, right? We think about how we have products and reactants. We could graph them, how much energy they have, and we understand if it's exergonic, reactants have a higher energy, products have a lower energy, right? And ideally, right, you would just cash out the difference in energy and there you go. You've got it in your hand. It could be released as heat. It could be used for another reaction, right? And that would be great. That's thermodynamics. All it cares about is the products and the reactants, how much energy you have. Everything in between those two values will be released or absorbed if it's endergonic. Activation energy has nothing to do with thermodynamics, right? Activation energy is kinetics, but it kind of fits on that graph, right? What we really see if we start out with high reactant energy, yeah, it wants thermodynamically, it wants to make low energy products, right? It will release energy. That is favorable. It's spontaneous, but it can't just snap its fingers and become products and release energy. The thing that comes into play with activation energy is we have to spend energy to get energy, right? And we, you know, you were talking about climbing up a slide to slide
yeah, we
down.
were talking about this before.
That makes sense. I was thinking personally, as someone who loves roller coasters, I like roller coasters, right? You start at, you know, a given point, right? A given elevation. In order to get a really big drop, you have to go up first, right? And that requires energy. Ultimately, if we were thinking of, let's say, potential or gravitational potential energy, right? If you gain elevation, more and more energy, right? You have to expend energy to get you up high. And then you gain all of that back, right? You drop further than you started, energy is released, right? But you have to spend energy in order to get that payout. So again, activation energy is how much energy you have to invest to essentially convince the reactants to start becoming products.
Right.
I think it's important to recognize that, again, reactants don't just snap their fingers and become products. They have to morph into those products.
Right.
And there comes a point that you are like part product, part reactant, and you're like actively switching between the two. That is incredibly uncomfortable for a molecule to be halfway between two different molecules.
Right.
And that's called the transition state.
Yeah.
So it's basically activation energy is how much energy do I have to invest in order to get that transition state that's so uncomfortable to occur. Once it's there, it can break down into the products and you get a net release of energy. We like that. But sometimes that activation energy is extremely high.
Extremely large. Yeah.
And it comes back to something we were talking about last episode with thermodynamics, that you can have a reaction that is incredibly favorable, like releases so much energy, and yet it will never happen. It's favorable. It would in theory, but kinetically, it is so slow because that activation energy is so high.
Yeah.
Yes, it's so uncomfortable to go from reactants to products. So we could see it again, see it on the thermodynamics diagram for let's say an exergonic reaction, but I want to stress this is not a thermodynamic concept. It has nothing to do with delta G or KEQ. This is kinetics land. But it's useful, I think, to see it on that visual, right? Since that's what we think of as like the reaction diagram.
Absolutely.
Right?
Yeah,
So,
absolutely. That like mountain that you have to go over. And
Yeah.
right, imagining that bigger that mountain that you have to go. I'm sure like anyone listening to this podcast, I think most people have seen that reaction diagram, at least at some point in their scientific careers at this point. But recognizing that that hill that they have to overcome is that activation energy. But again, has nothing to do with the reactants or products. But yeah, higher the activation energy, slower the reaction. You decrease that activation energy, the reaction is likely to go much, much, much faster. Much faster. And again, we'll talk about later what actually can do something like that, decrease the activation energy. But kind of those two factors that impact that little k constant from that Arrhenius equation. So we have our rate constant, but then where we see our rate constant is when we talk about our rate laws. Now, the rate laws is if we actually want to calculate the rate, you'll see rate equals k times some substrate concentration to an exponent. Because here introduces the third thing, because we've talked about two factors that can impact the rate. Activation energy can impact rate, temperature can impact rate. But thirdly, and this is an asterisk, sometimes substrate concentration can impact rate. And notice I said the word sometimes, because it's like a lot of times we're like, oh, it always impacts. If I increase the substrate concentration, it'll always increase the rate.
It has the capacity to.
It could. but
Yeah.
not always, right?
Yeah.
So when we talk about rate laws, the MCAT generally focuses on like 3 different like rate laws, right? And so honestly, Molly, I love how you explain these. Like when we were talking about before, I was like, I'll leave you to explain these
Okay.
ones.
All right. I got it. Let me cook. So
Right, you cook.
as you said, right, when we talk about rate, if we want to calculate how fast a reaction is occurring, right? Let's say in reactions per second, rate is going to be equal to K, that little K that we just spent so much time talking about, right? With temperature and activation energy taken into account, that weird E thing that you're probably never going to calculate because that calculation would be kind of gnarly, right? Understand that conceptually. If rate equals K times possibly some substrate concentration. What I like to say is always assume rate is equal to K times substrate concentration raised to an exponent. Okay. And there could be multiple substrates in there, right? It could be substrate A times substrate B. That's fine. We'll talk about that too. Okay. So we talk about zeroth order, first order, and second order. Those are the ones that we talk about most frequently with the MCAT. What I want you to know about these is the number associated with it, 0, 1, or 2, is the sum of the exponents. Okay, so let's consider zeroth order. Rate is equal to K, little k, times, let's just say substrate A raised to the 0. What is something raised to the 0 going to be equal to?
Always going to be 1, no matter the number.
Yeah, 10 to the 0 is 1. A million to the 0 is 1. Negative blah, 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 to the 0 is 1. It's always going to be the case. So if we have rate is equal to K times substrate concentration to the 0, what that means is I could change the substrate concentration to whatever I want. I could increase, decrease. I could do whatever I want. It's never going to change the rate, right? Because it's not even a component of my rate law anymore. It's rate is equal to K, basically.
At that point, right?
Yeah, because the substrate concentration is raised to the 0. Now, if we consider a first order reaction, again, rate is equal to little k times substrate concentration, in this case, raised to the 1. Now, if you raise something to the 1, it's just going to stay the same. 10 to the 1 is 10. A million to the 1 is a million, right? So what this is saying is rate is going to increase with A. And this is going to be a linear relationship, right? Because K is a constant. Kind of like Y equals mx plus B, right? It's Y is the rate in this case, and we have a constant times that
a variable,
right? And it's raised to the 1, right? Y equals mx plus B. It's just a line,
right?
Okay. If you increase the substrate concentration, the rate will increase, right? The slope of the line would be equal to your K, which is a constant. It's not going to change. Now, second order kind of has a couple options, because what it means is the total sum of those exponents is 2, right? So we have rate is equal to K, and it could be times substrate A raised to the second power. It could also, though, be rate is equal to K times substrate of A raised to the 1 times substrate B, or concentration of substrate B raised to the 1. That is also a second order reaction. That being said, when you plot it, right, regardless of how you set it up, if that total exponent is 2, as you increase the substrate 1 unit, the rate is going to be squared.
Exactly.
Right? It's proportional to the substrate concentration squared. So you're going to see an exponential function instead of a line, right? Again, zeroth order is a flat line. You can do whatever you want to substrate concentration. It will not change. Is it substrate concentration raised to the 0? If we have substrate concentration raised to the 1, as in a first order, you're going to get a line, right? Typically with a positive slope, because the K is positive. And then you're going to see second order where you have that exponential increase. That's the fun part. Don't we love rate?
We love rates, right? Yeah, and I love that when we talk about rate laws, because they really describe how much does the substrate impact the rate. Does it not impact it at all? Does it impact it linearly, or does it super impact the reaction? And something I know I get asked all the time in CN classes, like, how do we determine the order of the reaction? Like, we have this rate law and we have these exponents, like, how can we determine the order? A mistake I see all the time is that we just assume the coefficients, right? If I have a reaction, 2A plus 2B goes to C, that's my reaction, right? Can I simply say that the reaction is second order to A and second order to B? Absolutely not. Right.
That,
And
to clarify, you could do that for KEQ. That would be the exponent for KEQ,
yeah.
but that's a different land than
We're in a
rate.
different
Rate is
world
kinetics,
right now.
different world.
Yeah. So, and this is a really important thing where you cannot use the coefficients to determine those exponents, the rate order. You have to determine them experimentally, right? Where you would need to do what you have is you have a reaction, you see the rate, if you have hold the concentration of A and B constant, and then you double one of them and see how the rate changes. Okay, double the other one, see how the rate changes, and experimentally you determine the order in terms of each one. But a mistake I see is that we just assume it's the coefficients.
To clarify with the data, right? You'd see this in a table. You'd see one column with the rate, how fast is this reaction happening? And then, like you said, you'd have the another column that says the concentration of A and another column that says concentration of B, right? These are kind of interesting to do experimentally because you'd want to find one that doesn't change A, but that changes B, right? And then you want to see how does the rate change if it does. If you double A or double B and A stays the same and your rate doubles, cool. Your first order with respect to B. If you double B and rate becomes 4 times larger, that's 2 to the second, right?
Second
Be second order with B. Right. Yeah. So you'd have to go in and decide which ones you can compare. I always like to clarify that because people are like, where can I find a practice question on there?
Right.
They're in the AAMC material. We have them on our website, but that's how you'd recognize it. If you see a table where you have rate and you see substrate concentrations, they could be asking you what order the reaction is.
Right. Right. And I think this is also really cool to talk about when it comes to multi-step reactions, because this is also important when talking about rate, because say we have a reaction that has multiple steps.
Oh, right? Now we're getting complicated.
Right now we're getting kind of tricky, but I don't want to go too much on this. But
yeah,
when we have a reaction, well, when you have a reaction with multiple steps, the question that has to become is which is the rate determining step?
Yes.
Right? Because if you have multiple steps, one of them has to be the one that determines the rate. I mean, they don't all determine the rate. The one that determines the rate is the slowest step. The slowest step is always the one that determines the rate. That's the one we call the rate determining step. And now what's really interesting when it comes, and this is the why behind why you can't simply use the coefficients, right? You can't use the coefficients because you don't know if a reaction is one step, if it's multi-step, if all of the reactants actually contribute to the rate. And so this is why it has to be determined experimentally, because you need to know what the rate determining step is. So I know that's just a term that students would have heard before, like what the rate determining step. If you have a multi-step reaction, it's just the slowest step, because again, it's all about speed. The one that determines the rate is just the slowest one.
Yes. Yeah. I love that you bring that up because that is such a beautiful connection to things that we will talk about in future episodes. When I hear rate determining steps, I'm like, of glycolysis.
Oh, we're gonna, oh my God.
PFK1, what?
Yeah. And which, again, if you heard the word glycolysis, hint, hint, nudge, nudge, what might be coming. But yeah, and actually, and actually, I think this is kind of a really great place to kind of talk about. I mean, I don't think I have that we need to touch on anything else before we talk about the next kind of big thing, which is really related to metabolism, which is enzymes. All right. I mean, we both love enzymes because you cannot talk about rate without talking about the thing that can really impact rate, which is enzymes. Now, let's kind of first talk about, like, Molly, what is the thing with enzymes? What's like the main thing? Like, what do enzymes actually do?
Yes. So before we say what they do, they're all proteins. Okay. Sure. They're proteins that help us speed up reactions, which means they are kinetic beasts. They live on the kinetic world, right? I always call them kinetic beasts. I don't know why.
That's wild. Like I've never heard that.
I picture them of like beasts of burden. Like they're just helping us, like, you know, like a horse, a mule, like they're just helping us do things more efficiently, more fast, you know, like enzymes are kinetic beasts. They are made of protein, of course, and their job is to help us speed up chemical reactions, right? That means it's a catalyst, which means it must increase the reaction rate. How does it do that, though? We now have all the language to discuss the reaction rate. We said rate is equal to K times the substrate concentration. I will tell you right now, it's not going to change substrate concentration. This is kinetics, right? It doesn't concern itself with that. What an enzyme will actually do is help that transition state going from reactants to products be stabilized. It's basically a physical, like a warehouse that this reaction can take place in. You have the reactant come in, right? And the enzyme is helping facilitate it beginning to become a product. Okay. In doing so, it is stabilizing that transition state. That's the halfway point, or the, point where you're
That
part
uncomfortable
reactant.
place, right?
Yes,
Yeah.
exactly. Where you're like half product, half reactant. And in doing so, keep in mind the transition state energy and the difference between the reactant and the transition state energy, that's activation energy. If we're bringing down the energy of the transition state, what we're doing is minimizing the activation energy. So how then is that going to affect rate, right? In our rate law expression.
Absolutely. I'm trying to think about like where all my variables plug in, right? On my rate law expression. Well, I know rate equals K substrate concentration. Well, we just said doesn't impact my substrate concentration. Well, the only place else it impacts is K, the little k that we just talked about from that Arrhenius equation, because it impacts the activation energy. Remember, we talked about the variables that impact rate. It really hits on that one, the activation energy. We decrease that activation energy. And again, you'll understand this if you look at your Arrhenius equation. It will increase your rate constant. I know seeing that in the Arrhenius equation can actually be kind of tricky because it's a negative exponent, but what you need to know conceptually is decreasing the activation energy always increases the rate. And that's how enzymes speed up the reaction.
Yes,
right?
Absolutely. And it's important to, again, mention, I feel like we can't say this enough. Thermodynamics land is on one planet, kinetics land is on another. These belong in kinetics land. Right. So we could think about the sort of variables that we sort into each one of these different planets, right? When we talk about KEQ, delta G, delta H, delta S, those are all thermodynamics. An enzyme will not change any of those things. They will not change any of those things. They will also, again, they won't push equilibrium. They won't change equilibrium. What they can do is change reaction rate. Okay. They stabilize the transition state. They make it lower energy. So activation energy can go down, rate can go up. That is the extent of what they can do.
Right.
And again, it's an important factor with just bringing the necessary reactants into the same spot and allowing them to react together, right? We talk about how, if you want, like you said, 2A, 2B makes C. Okay, they're all kind of binding together to create this product. If they're miles apart,
right,
it's going to be really hard for 2 of the A's and 2 of the B's to join and make C, right? It's just physically, if they're really far apart, the reaction's not going to happen. Even if it's favorable thermodynamically, and even if the activation energy is pretty low, if they're physically super far apart, they're not going to bump into each other and react. So an enzyme, in addition to stabilizing that transition state, is also helping by just making sure all the key players are in the same place at the same time.
Right.
Yeah. And so that's a very important factor here too. It's bringing things together so the reaction can occur. And we could talk about other interesting factors of enzymes, or things that enzymes could theoretically do that are based on this proximity, right? We won't get into a ton of detail with this today, although we'll hit on it later. Another cool thing enzymes can do, again, reiterate, they do not change KEQ. They do not change delta G. One thing they can do though, and we mentioned this yesterday.
Like last episode,
yeah,
right?
Not yesterday, last episode, right? There are reactions, most of our synthetic reactions, making a protein, for example, those are all non-spontaneous. They will not happen, and yet they do. And this comes from taking a spontaneous reaction and a non-spontaneous reaction and allowing them to work together, right? You take the energy released from a spontaneous reaction and you use it to fuel the non-spontaneous reaction. Same deal as we were talking about before. If reactants are incredibly far from each other, they will never react. If 2 reactions, one spontaneous, one non-spontaneous, are really far from each other, how will that energy be transferred? It can't.
Right.
So enzymes can, not all do, but enzymes can also carry out what we call coupling.
That idea. I've been like waiting for that word. I'm like, oh, you're like saying it. Yeah.
Like if we thought of the warehouse, right, allowing in the reactants and helping it transition to the products, we're basically allowing multiple reactions to happen at the same enzyme. And we're taking the energy from one and just scooching it over and giving it as a reactant to the other, right? It's the same deal with proximity, but in this case, again, I want to highlight, it is not changing the delta G of either reaction. It is simply varying or moving the energy from one reaction to the other because they all take place on the same enzyme.
Right.
So
Right.
enzymes are awesome.
So cool, right?
They're so cool.
Basically, yeah, they create this really great environment for the reaction to help facilitate that reaction, to really stabilize it, make everything great in order to get to the products. But again, yeah, really stressing, if it started out non-spontaneous, if this reaction wasn't going to happen to begin with, an enzyme's not going to change that. If it was non-spontaneous to begin with, sure, we can decrease the activation, sure, we can make a great environment, but it won't make a non-favorable reaction favorable. It just makes this environment this really optimal environment for the reaction to occur, possibly coupling, making this, maybe the pH needs to be right. Maybe they need to orient the substrate a certain way. That's what they do. They really facilitate it. And that place that they facilitate it, right? It's a really important place on the enzyme known as the active site. Now, this is going to be very important in our conversation about some kinetics later on, but the active site is that, I like imagining that's where the action happens on the enzyme. Because if you visualize enzymes are these big protein balls. Like you said, they're proteins. So many amino acids, they could be huge. There's one place on that enzyme known as the active site. And that's where kind of the catalysis occurs. There's not to say that there's not other very important sites on that enzyme, but the active site is where we really facilitate that reaction. I mean, so we generally refer to the substrates entering the active site, binding there in order to be catalyzed. But we also have binding sites on other parts of the enzyme, because when we produce enzymes, not all enzymes are just produced, ready to go, activated, they're like chugging away, right? We want to be able to regulate our enzymes because we can imagine, right? Like, I don't want all my enzymes always active all the time, right? That might actually be a little bit problematic. I won't be able to respond to, actually, I'm sure there's lots of pathologies that result as a result of enzymes active all the time. So we want to be able to regulate our enzymes. So a lot of times we produce kind of an inactive version of our enzyme, which is known as an apoenzyme. And this apoenzyme is like, we produce, again, there's nothing wrong with it. It just isn't activated yet. But in order to become activated, it needs essentially a friend. It needs something to tap it on the back to kind of bind to it in order to make it whole, right? And so this kind of thing that binds to our apoenzyme is known as a cofactor or coenzyme. Now, I like to kind of distinguish what it means to be both. So the broad term is a cofactor. A cofactor is just anything that helps the enzyme to become whole in order to function. Now, a cofactor is kind of the broad umbrella term, but we have certain cofactors that are organic, right? And what do I mean by that term organic? Organic is just, and I'm not talking about the Whole Foods organic, that's a totally different thing. I'm talking about the term organic, and when it comes to like chemistry and biology, which means contains carbons, right? If we have a cofactor, something that helps my enzyme to work, and it contains carbons, therefore it's organic, we call it a coenzyme. Right. A coenzyme is just this organic cofactor. It doesn't mean a coenzyme is not an enzyme in itself. Right. I want to make that very clear. It's not an enzyme. It's just a cofactor that's organic. Right. And we have a bunch of examples of these. Like NAD is a great example of a coenzyme. Something that's organic helps FAD. Great example. And again, these terms are related to metabolism. So hint what's coming.
Enzyme A.
Enzyme A, right?
Q.
Right. All of these things are very important because when they bind, they allow the enzyme to actually work. So when the cofactor coenzyme binds to an apoenzyme, it becomes whole, or a holoenzyme. That's how I kind of remember. It's whole since the holoenzyme, and now it's active. It's ready to get going. But we notice there are these. The cofactor coenzyme doesn't always. It's not always. Not even usually binding to the active site. It's usually binding to some other place on the enzyme. Because remember, we have this whole huge protein ball of an enzyme. There are binding sites other than the active site that these cofactors, these coenzymes, can bind to. And it's important to recognize that there are these other really important sites when we talk about kinetics. But yeah, and so I think kind of just really recognizing, okay, not only what the enzyme does, but what even is this ball of catalysis that we talk about?
Yeah, so I love that you bring up how big an enzyme is, right? It's kind of like the genome, right? The genome is vast, like unfathomably large, and yet only a small portion of it is actually used to make protein. Right. It's only a small portion will be used to make, well, I guess I should say will just be transcribed. Sometimes we like our RNAs as it is. And a lot of people think, oh, the rest of it is just junk. It's like, why would we just accumulate junk? Right. There are a lot of important things that the rest of the genome does. And it's the same way with enzymes, right? An enzyme is this giant ball of protein. There's a small area where the action happens, like you said. That's the active site. It's where the substrate which is just, the reactant that we're working
Sure.
with, is physically going to bind. That would be the binding site within the active site. And then we have the catalytic site that you actually have the chemical reaction happen, right? So the active site is, like you said, where the magic happens. That leaves the entire rest of the enzyme. Having small changes in the rest of the enzyme can also drastically affect the function of the enzyme, because the rest of it, other than the active site, is still critical, right? When we talk about biomolecules, structure and function go so hand in hand. If you mess with the structure of an enzyme in particular, but of any biomolecule, usually the function will change.
Absolutely.
There are different types of mutations that we could talk about in terms of gene expression, silent mutations, frame shift mutations. We're not going to get into that today, but what I want to hit at is, just because you're not mutating or messing with the active site does not mean that you're going to keep the enzyme functioning. Like you said, we definitely have sites that we require cofactors and coenzymes if the enzyme is produced in an inactive form. Again, apoenzyme plus a cofactor or a coenzyme, if it's organic, create that holoenzyme. It's the holoenzyme, right? But the rest of the enzyme also has important spots. When we talk enzyme inhibition, right, we really need to make sure that we have that understanding, because most types of inhibitors, all except one, are going to bind somewhere that is not the active site. The cool thing is, if I bind somewhere that's not the active site, because structure and function go so hand in hand, that actually can change the three-dimensional shape of the enzyme to change the affinity between, or the desire to interact between the substrate and the active site. So we need to pay close attention to what is happening elsewhere on the molecule, right? In that same vein, we could think about how enzymes interact with their substrates. A lot of people think, and it's logical to do so, that enzymes accept their substrates like a lock accepts a key, right? The key just fits perfectly into the lock. Enzymes and substrate interactions are very specific. Typically, key perfectly fits in the lock. You turn it, reaction happens, we're happy, right? Door unlocks. But like we talked about, structure and function are so important, right? If you take a substrate and start to bring it closer to the active site, let's say the substrate is super negative, right? Maybe the active site is super positive. As they come closer, those charges are interacting, right? The actual enzyme is going to change configuration to kind of grab onto that substrate. It's a very active process. It's not a perfect, right? My solid key fits
lock
into
and key
determined lock, right? I always picture it like a handshake, right? You stick your hand out straight, you reach it towards someone, and as your hand gets closer to their hand and interacts, you grab onto it and you hold, right? I always, and it's every time I feel like it's silly when I say this, but imagine you got a job interview, or you're closing a deal and you reached out with the exact confirmation that your hand would have to actually shake someone's hand. Imagine reaching out with this weird little claw to shake someone's hand after sealing a deal, right? It's not a lock and a key. It's an active reorganization where you grab onto each other. That's the way that we have enzyme substrate interactions. We call that the Induced-Fit Model, right? Because the fit between the substrate and enzyme is induced or created by proximity. It's not lock and key. Because it's not that simple. As a heads up, pretty much nothing in biochemistry is
ever that simple.
It's
we try to simplify a lot of times, but it's like, oh my goodness, it's so much more complicated than a lot of times than we originally thought. But that's just, we talk about, the reason we even still talk about the lock and key is because I always like, and why we talk about people like Freud, super psych soc, is it's always important to know where we started to understand where we are now. Psych social is another day entirely, but I have so much strong opinions about Freud, as many do. Why would we even talk to him? Why do we even talk about him still, kind of deal? But and I think it's also important to know, like, we now know like what the enzymes, like how they actually do what they do. How do they actually speed up a reaction? We know that activation energy. We know the structure of an enzyme, the different components of an enzyme. We know how it combines with the substrate, but it's also important to know that we have how we classify enzymes. Because enzymes, right, we classify them based on what they do. And I think that's so intentional about, like, when we look at, and this is, again, why again, we're always about intentionality. Again, I said the word, we have to say the word every single episode.
I know we do. And we need to talk about the fact that we've said it every episode.
Every single time, because we need to know that there's an intention behind all of these enzyme names, because we classify them based on what they do, essentially the reaction that they catalyze. Because like we said, right, all it does is decrease the activation energy, but we also just talked about that it's very specific. Not all enzymes can can catalyze any substrate. Because again, that induced fit model has to be just right in order to kind of fit and induce. So specific enzymes are needed for specific types of reactions. Now, when we talk about the different types of reactions, because there's this kind of mnemonic that we use.
Yes.
I'm not a huge mnemonic person, but this is a really good mnemonic, which is LIL HOT. LIL, LIL HOT. I don't know. It's just a little hot. You know, it's just a little hot.
It's just a little hottie, no problem.
LILHOT.
Yes.
And so I
love it.
And again, I remember I only learned about LIL HOT like I think I learned it from I think I had one of your presentations and I saw LIL HOT and I was like, what is this? I always knew what the class was. I think that's super, super cool. Well, LIL HOT describes the different classifications of enzymes, but it's based on what type of reaction they catalyze. So we can kind of go. I'll take the first three. You can take the next three if you want. I'll take LIL. You take HOT. So again, I don't know if there's a correct first L. I actually switch it up whenever I teach it just for fun. But the first L in LIL HOT represents a lyase. Okay, I could have used either one. So a lyase is an enzyme that catalyzes a reaction that you cut a bond without the use of water. Now, this is important. When you like a lyase, that's essentially what it is, an enzyme that catalyzes a bond breaking without water. Now, notice what I'm stressing here. Without water, right? Because we have a different enzyme, which you'll take, Molly, that uses water to break a bond. Now, when we see, there is different cases that we see these kinds of reactions, but if I were to think about a lyase reaction, it's actually the one I think about usually actually isn't a great example because I usually think of like a synthetase.
I always think of a decarboxylase.
Oh, that's a good one as well. That's a good one.
I'm like, that's really the only time that you're gonna see it.
So frequently, yeah. And then the one I think about is technically a lyase, but it's an awful example of a lyase because it's doing the opposite. But a decarboxylase, I think, is actually a way better example where it's cutting off a CO2 from a molecule without the use of water, right? Because if you have a carboxylic acid, you can remove that carboxylic acid in order to produce CO2. That reaction is a decarboxylation, right? And that decarboxylation, you're cutting a bond, removing that carboxylic acid. You are not using water for that cut. Because you're cutting not using water, it's a lyase reaction, or it's a reaction catalyzed by a lyase. Okay, so that's, I think about that one. That's a great one. Don't think about my synthetase situation, even though technically it is. Technically it is. Don't even think about that one.
Extra points if you Google it and write about it in the comments for other people to learn. Right,
because when you talk about enzyme classification, I don't even want to dive into that right now. Like every enzyme has its own classification. You can even look it up. I remember when
that's what we're sharing, by the way. We're not sharing like this is the only name it could ever have. There's a million names that could fall under these. These are what are the classifications of the enzyme based on the type of reaction it is.
Right.
Okay. Lots of different options for naming, but these are the classes. You are one or the other.
Exactly.
Lyase is the first.
Right. And then the second one, right? LIL is an isomerase. Now, when we talk about, again, I love the names because they tell you what to do. I think of isomerase. So all it does is take a substrate and turn it into an isomer of itself. But I say that word isomer, and I feel like I need to define it. An isomer is simply a different molecule with the exact same molecular formula. That is the definition of an isomer. So if I'm changing a molecule, but I'm not changing the molecular formula of a molecule, am I adding or removing anything to said molecule?
Nope, same atoms.
Same atoms. It's the exact same atoms, but it just has a different orientation or a different way, a different orientation of its molecules. So it's all about transferring functional groups within the same molecule. It moves functional groups around in the same molecule. It changes the structure, but does not change the molecular formula. If it's a different molecule, like glucose to fructose, they're both C6H12O6. Absolutely, same molecular formula, but it's a different molecule because the orientation of the atoms are different. So those are isomers. So we have different examples, but I think the main isomer I think of is a mutase, or isomerase is a mutase, right? Where we have like phosphoglucomutase, glucose 6p to fructose 6p, changing glucose to fructose. But again, it's not adding or removing anything. It's simply just changing the orientation, moving functional groups around within the same molecule. Okay. Again, I want to stress the within the same molecule because there's another enzyme type that is not, but it's within the same molecule. The last part of LIL, L-I-L, the last L is a ligase. Now, a ligase is an enzyme that joins things together, that forms a bond between two larger groups. If you're forming bonds, you're ligating those groups together. And now when I think about a ligase, an enzyme that is classified as a ligase, I think of DNA ligase. That's probably like the main one for the MCAT. Like you think ligase, think DNA ligase. That's the thing that, when you're doing your DNA replication is joining those kind of bits of DNA together, right? Especially on that lagging strand with those after the Okazaki fragments. So it's joining DNA fragments together, joining these two groups, forming a bond there, ligating those bonds. So ligase, think of an enzyme that forms a bond between two groups.
Yes. Okay. So LIL, you did lyase first. You could do ligase or lyase first. It doesn't matter. Because they're all classifications, they're all equal to each other, right? Like you are one or the other. There's no hierarchy, right? So lyase, isomerase, ligase, right? Now we're getting into the hot. We got the hot stuff.
The hot stuff. And I actually think like the hot stuff, like you'll see way more often than the little stuff. Like I just think, especially with the first and second one of hot, I think those are so, especially the second one when we talk about what we're going to talk about, so often you see them.
Yeah. All three of them really, right?
Yeah.
So to give you a fast rundown, hot is hydrolase, oxidoreductase, and a transferase. These are everywhere, and a lot of them go by a lot of different names. Okay, let's start with hydrolase. When in doubt, if you see a bond breaking and you don't see anything special, it's probably a hydrolase. Okay, a hydrolase is going to undergo a hydrolysis reaction. We take water and we add it to a molecule in order to break a bond. Remember back when Mike was talking about a lyase, it breaks a bond without water, right? This is a bond that breaks with water. Okay, so a hydrolase is using water to break a bond. The reason why I say when in doubt, it's probably a hydrolase if you see a bond breaking and nothing else special going on, is because if we talk about any of our macromolecules and how they're polymerized with what we call dehydration reactions. You take two things and you smush them together and water is produced. If you want to undo that bond, and say you took 2 amino acids and you underwent a dehydration reaction to make 1,2 residue peptide. If you wanted to then break that back into 2 amino acids, you're going to have to do the opposite. You formed water to bring them together. You have to add water to break them. Okay. If we think any sort of protease, and there's so many that we learn about, there are so many proteases that we learn about. If we think of a lipase, right? If we think of a any sort of bond that breaks or enzyme that breaks down sugars. So like amylase is a good one. I wish there was like a carbohydratase.
Yeah, carbohydrates. Yeah, I don't know.
Yeah. But anything that breaks down any of the macromolecules, including DNases and RNases, right? Those are all hydrolytic enzymes. If you break down a biomolecule and you don't see redox and you don't see anything special, it's probably a hydrolase. Statistically speaking, you learn so many of those. So you're adding water to break a bond.
I think most of the digestive enzymes from the pancreas, for example, all hydrolases. And then that's all just breaking down. So yeah, and that's why I'm like, when I hear hydrolases, there's like so many. You're so likely to see those on this exam.
Yeah, and the lysosome as well. All the intracellular enzymes as well.
Right, so many.
And I feel like it's important to have talked about protein structure a little bit with enzymes because, again, these are hydrolytic enzymes are examples that you do not want these active unless they need to be active because these things break down all of our biomolecules. If you let an activated protease loose in your cell, you will digest yourself, right? When we talk about digestive system, we very much excrete these, right? or secrete them into the outside of the body, right?
Mhm.
Which the GI tract technically
Technically, yeah.
is. All of those are activated by the conditions under which they should be active, right? I don't want this loose in the cell because it will wreak havoc, right? It will only be active, let's say, in the low pH of the stomach, right? There's some activating step, kind of similar to what we talked about earlier with enzymes, right? They're not produced in an active form. In the case of digestive enzymes, sometimes they're actually produced with an extra chunk, like an extra leg, so to speak, that gets cleaved off, but we'll talk about that another time, right? Hydrolase, vast majority of the enzymes that break bonds, right? And they use water to do it. That's why it's hydrolase, right? Carries out hydrolysis. The O is an oxidoreductase. And this, I feel like we barely even need to explain because of the background we've set.
Oh my goodness, yes.
Oxidoreductase does oxidation and reduction. So this is an enzyme that helps catalyze a redox reaction, a transfer of electrons. We spent the last 2 episodes talking about
Really
redox.
hammering it.
So you guys know this one already, right? A redox reaction is catalyzed by an oxidoreductase. So if you see a transfer of electrons, there you go. Those also tend to have pretty nice naming, right? Oftentimes you'll see oxidase. Sometimes you'll see oxidoreductase. Sometimes you'll see dehydrogenase.
I mean, that's when we talk about metabolism, that one is going to come up so often.
Yes. So you'll see all things like that. That's an oxidoreductase. Again, we've talked about them in the past. We will continue to talk about them in the future.
Of course, always.
The last one is a transferase. And this kind of hits at what you were talking about with an isomerase. Isomerase, again, like glucose to fructose, it's the same atoms. It's simply rearranging them. A transferase is an enzyme that takes a functional group or an atom from one molecule and now brings it over to another one, right? It's the same idea as redox, right? We're taking electrons and bringing it here. But instead of taking electrons, we're taking whole atoms, right? I'm transferring, especially functional groups, this one from here to here. Right. If we think of a kinase.
Great example. Yeah.
Yeah. We have ATP, energy, and we are taking one of those phosphates and we're sticking it onto something else. A kinase is a great example of a transferase because it's all about just moving functional groups from one to the other. That wraps us up. So LIL HOT, lyase, isomerase, L ligase, H hydrolase, O oxidoreductase, T transferase. Whew.
Lots of classifications.
Enzyme classifications.
Enzymes are so much fun in that way. But again, I really like them because I definitely heard this before. I don't want to memorize these, right? I don't want to memorize what an oxidoreductase does because the name tells you what it does. And so I think LIL HOT is a great mnemonic to kind of, okay, what are the letters to kind of help you out with that first one? But yeah, I love enzymes for that reason because they're named so intentionally, like named for what they do, like 99% of the time. Are there a few examples where sometimes the name is very misleading? Absolutely, there is. I know, and we'll talk about that. And I actually love talking about that one. But anyway, we'll talk about that one relatively soon. But yeah, I mean,
if you haven't picked up on it yet, somewhat soon we will be walking our way into metabolism, which will be super fun. We've got a couple other things we need to talk about first before we're ready to bite that off. But metabolism will be a fun one and a big one for us.
Right. And actually, I think I'm looking at how long today's episode is.
I know it's going to be long.
It's this one. This one's a long one because I think why don't we kind of keep the listeners waiting, and then next week, why don't we kind of talk about. This is kind of enzymes and kinetics part 1 because there's a huge topic. I think it does deserve its own episode on.
I know.
Yeah.
I was thinking about that.
Yeah.
Enzyme inhibition is critical. And we haven't talked about kinetics yet either.
But the Michaelis-Menten kinetics.
Enzymes.
Yeah. And then I just said the name and somebody was like, oh my goodness, no, not that word. But yeah.
Lineweaver-Burke.
Right? So
My favorite.
So I think we'll probably let's kind of say this is part 1 of enzymes and kinetics because we kind of this is all the foundational stuff about talking about kinetics, what kinetics is, how it relates or doesn't relate to thermodynamics, and then enzymes. And the next episode, what we're going to be hitting on Michaelis-Menten kinetics, what that even is, how they got those plots, what they tell us Lineweaver-Burke, how it extends, and then we'll hit on inhibition, the different types of inhibitors, and how to understand them and not just memorize what happens when, X inhibitor is present kind of deal.
Yes. I am so excited. I mean, it's kind of a similar situation to the thermo and electrochem episode that we just finished. It's like electrochem is such a big, scary topic, right? I think enzyme kinetics and enzyme inhibition is such a scary topic, but they really don't have to be if you lay the right groundwork, right? What we've done is lay all the important groundwork, right? Again, recap, we talked about what rate is, right? We talked about the rate constants, the things that influence them. We talked about activation energy. We talked about how to decide if something's first, second, or zeroth order and what that really means. We introduced enzymes, talked about their structure, the different parts of an enzyme, what it can and cannot do, especially given that enzymes are kinetic and not thermodynamic, right? We talked about the limitations. We talked about the different types of enzymes, right? We are primed and ready. Once we hit this material, again, it's springboarding us into all of those really fun enzyme kinetics discussions. And then once we hit inhibition, we're free to make our way through metabolism.
Oh my goodness, because we've done all the legwork, right? Once we actually get into metabolism, now it's like we're gonna be seeing, and I think that's why the MCAT loves metabolism, because like we've said before, anytime they can combine areas, anytime you can combine different subject areas, the ideas of equilibrium, Le Chatelier's, right? The ideas of like different, like greater metabolic processes. Anytime you can put those things, they love talking about them in those passages, and metabolism is a great example of that.
We'll even see some things, smaller metabolic processes that affect bigger body systems. Right. Like we'll tie back, I'm sure, to blood and respiration, bicarbonate buffer system, all those fun things.
All those things are related, right?
Metabolism is going to give us a chance to really make all this material shine.
Yeah. But again, this is just part 1 of Rate Enzymes. Join us for part 2 episodes where we really lock down those like really high yield concepts of Michaelis-Menten, kinetics, and enzymes and inhibition. So I'm excited, Molly.
Yes, I know. Me too. Super exciting. And I mean, all of this material is high yield, but next week or next episode, get ready to rock and roll. We're going to be working from minute one.
We're rocking and rolling.
Yes. Right. Thank you guys for listening to our first part of enzymes and kinetics. And we can't wait to catch you in the next one. Until then, happy studying. Have a good rest of your day. Make sure you get sleep.
Or night, if you're listening at night.
Or night. Yeah. Maybe if it's nighttime, go get sleep right now.
Go to bed.
Go to bed. Yes. All right, you guys. Bye, everyone.
See you, everyone.